Okay, so we are finally done with plants and moving into animals. So this is the lecture for chapter 30, an introduction to animals, and we're going to briefly go through the basics here of what makes animals a distinct group, and also some comparison to plants and fungi. This will set the stage for later chapters where we'll dive into more specifics about the different groups that we talk about in this chapter. So what is an animal? Now, we should have a good understanding of what a protist is, what a plant is, what a fungi is. And animals are going to be now a totally separate group. Animals originated from a single-celled protist, it's believed, and they're believed to have originated from a common ancestor. So all animals came from the same multicellular common ancestor, right? That's what it means to be monophyletic, having one common ancestor. All animals share the same kind of specific traits. So even though they're monophyletic, coming from one ancestor, they have lots and lots of differences, but they also have these similarities. So they're all multicellular. So if it's a single-celled organism, it can't be an animal. They all share multicellularity. All of the cells lack a cell wall, but they have an extensive ECM, or extracellular matrix. All right, and that basically provides proteins specialized for cell adhesion, communication, right? All kind of around the cells. They've got this matrix. They are heterotrophic, meaning that they obtain carbon and the compounds they need from eating other organisms they eat. They don't do photosynthesis. They don't absorb it across exterior body surfaces. They ingest food, they eat it, and they take in the nutrients they need. All animals are going to move, so they're increased locomotion. They're going to have at some point in their life cycle, may not be in the adult form, it may be in the larval form, some type of movement. And lastly, all animals other than sponges, so sponges are kind of like an outgroup here in the animals. We'll talk about what makes them animals, but they all have nervous system, neurons that communicate, and also muscle cells that contract and move. So they can change the shape of the body when they contract. So lots of shared characteristics coming for this monophyletic group here. Again, all other eukaryotes animals share their all of these characteristics. So there's lots and lots of different phyla of animals. We're going to just focus on the circled ones here. We're going to look at Porifera, Cnidaria, Annelida, Platyhelminths, Mollusca, Echinodermata, and Chordata. So out of all of these, the ones that are probably going to be the most familiar to you are just one group, the Chordata. The rest of them usually don't think of as being animals, but they are considered to be animals. Okay, so the origin of the animal phyla. So looking back here in terms of the key innovations here, what sort of things do scientists, biologists consider when they're looking at the events that took place in the evolution of animals? So fossils are really important. They're the only source of direct evidence of what animals looked like, ancient animals looked like. It doesn't represent all animals equally, however, and we're more likely to find fossils of things that were abundant and lived in areas where the conditions were right for finding fossils. So just because you find a fossil doesn't necessarily mean that that's the only type of animal that was in that area. It just may mean that it was the most abundant. But they are a direct source of evidence, and we can look and see different layers of where we find different animals, and that can give us an idea of their relatedness. Second one, comparative morphology. Takes a look at the unique characteristics and synaptomorphies, or unique relationships among different groups of animals. So we can look and see, what characteristics are common among groups of animals, what are unique, like you said, those synaptomorphies. We can look at the body plan of an animal lineage, and we can look at these data and determine where did these characteristics arise and where are they shared. Number 3, comparative development. We can look at evolutionary biology, patterns of gene expression, right? What arose first here by looking at the DNA. And along those same lines, comparative genomics, looking at the whole genome and looking at the similarities in genes and phylogenetic relationships. Okay. Additionally, we can look at the origins of the embryonic tissue layers. We're going to see that there's 2 layers or 3 layers of embryonic tissues. We're going to look at where muscle arise. We're going to see that all animals have some sort of actin and myosin, which are the regulatory proteins in muscle contraction in some way, even if they don't have fully developed muscle. We're going to see the origins of bilateral symmetry, cephalization nervous systems. These are all things that are really going to be the most advanced. The coelom, which we're going to see. Protostomes and deuterostomes. We'll look at that more in the next chapter. And also segmentation, which is going to become important when we look at the different phyla of worms. So like I said, animals are a monophyletic group, meaning that they share just one common ancestor. And when we look back at the most basal animal lineages, or the oldest, it's going to be the sponges, which are the phylum Porifera. And these really early animals are believed to be the original multicellular organisms. And there's lots of fossil evidence to show that. We find the earliest animals 700 so million years ago being the sponges. And also, there's information from the geochemical evidence in the rocks to show that sponges were the first animals to have been multicellular. So in terms of development, something occurs in embryonic development called gastrulation. And what gastrulation is, it's, originally you got your egg and sperm come together, and the first cells that arise are going to start to differentiate into these different layers, and then the different layers are going to go on to become various organs in the body. So in some animals, simple animals, there's only 2 types of tissues, and those are called diploblasts, and literally diploblast means 2 buds. And there's some that have 3 layers that form, and those are triploblasts. So in the diploblasts, these early ones, you've got just 2 layers, the ectoderm and the endoderm. And the ectoderm is going to become the outer structures and the skin, and the endoderm is going to be all the inside lining. Where we have more complicated animals, we end up with 3 germ layers, and humans have this, triploblast layers, where we have the ecto, the endo, and the mesoderm, and the ectoderm gives rise to the skin and the nervous system. The endoderm gives rise to the lining of the digestive tract, and the mesoderm gives lining to the muscles, circulatory system, and many internal structures. So, meso, M for middle, M for muscle is generally how you remember that. Ecto is kind of a prefix relating to outside or skin, and endo meaning inside or the inner portion of the digestive tract. So when we look at the diploblasts, again, these are more simple animals, and the 2 groups of animals that are most commonly recognized as diploblasts are the ctenophores, or the comb jellies, and the cnidarians, which are jellyfish, corals, hydra, and sea anemones. So an anemone is basically just an upside-down jellyfish that's sessile, that doesn't move in its adult form. Okay, so muscle can be a little bit more complex, and when we look at different organisms, the ctenophores and the cnidarians, they don't have necessarily a muscular system the way we do, but they do change the shape of their bodies. And they also have a larval form that's going to move. So all animals have the genes for the contractile proteins actin and myosin. Ctenophores use cilia, but adults also have contractile tissue that can change the body's shape. Okay, so the idea that this process where these organisms that were not closely related independently evolve similar traits, having to adapt to similar environments, that's known as convergent evolution. So these organisms, even though we're not closely related to them, and some animals are not closely related to jellyfish and corals, they still had to evolve to have these similar structures because they lived in the same environment. So, the ability to move was not something that was unique to just one group in the animal kingdom. The proteins used in ctenophores and cnidarians come from the endoderm or the ectoderm, and we call them epitheliomuscular cells. These are functionally similar to the true mesodermal muscle cells that called muscle tissue, but they evolved independently as an example there of that convergent evolution. Okay, so that's kind of the origin of muscle tissue. As we've gotten more complex, now looking at the very earliest animals, being the sponges, there was no symmetry. So remember, think about symmetry is where you cut something and you end up with it being the same on both sides. It's a really key morphological aspect in a organism's body plan. And like I said, a body's symmetrical if you can, cut it in a plane and have, roughly the same thing on both sides. So cnidarians, like your anemones and your jellyfish, they have radial symmetry or spoke symmetry, which means there's at least two planes of symmetry. And if you cut like kind of along the center like a pie, you get identical halves. This was an independently evolved characteristic in this in their phylum, which is a kind of dermata, which we'll see, which has sea stars, sea urchins, brittle stars, all fall in that category. Now, bilateral symmetry or two-sided symmetry, that came later. So the really advanced animals, or the more advanced animals, I should say, have bilateral symmetry, which means you can cut it down the center and you have the same on both sides. So having bilateral symmetry indicates a more advanced body plan than radial symmetry. If you think about it, that makes sense because you got more going on inside of you if you're more complex, and you're not going to be able to cut 5 different ways and get the same thing on every side. The annelid word, which is showed in that picture, here is an example of something that has bilateral symmetry, one plane of symmetry running lengthwise down the middle. Okay. So along with, and I should mention too, that all triploblasts, or all of these advanced guys, are going to be bilaterally symmetric. So let's talk about the nervous system, because in the last few lectures when we talked about plants, right, we talked about how they did have a way to communicate. Right? They used hormones and things like that. We also use hormones, but hormones are slow. The nervous system is fast, and it can send impulses very quickly throughout the body. So biologists hypothesize that the evolution of the head and nervous system is tightly linked to bilateral symmetry. So we see that bilaterally symmetric organisms have a much more advanced nervous system. So it looks a little bit differently depending on the type of animal, because there's, again, very advanced animals and very simple animals. But in something that's like a cnidarian or a sea anemone or something like that, it's not going to have the nervous system, obviously, of a goat, right? So a nerve net is what you see in very simple organisms, where it kind of just is, in radially symmetric organisms, and it's all over. It looks like basically a net of neurons all around the body, whereas the central nervous system has neurons clustered together into more integrating centers, like a brain or a spinal cord or something like that, making up a central nervous system. So, it's really advantageous to have many neurons concentrated together. You can get into that cephalization, which is the evolutionary step towards getting a more complex brain. And you get from the central nervous system nerve tracts that carry information down throughout the body. So information can be sent from, out from the peripheral nervous system to the central nervous system and back and forth. So there's an example of a nerve net in a hydra, which is a little tiny organism in the jellyfish family. And you can see that the nerves are just diffuse all throughout the neurons. It's like a net. Whereas in a earthworm, which is more complex, you actually have a little tiny brain in there, a cerebral ganglion. Not much really higher level thought, obviously, going on in there, but you do get more so than the hydra. So the evolution of a central nervous system, an integrating center, came along, and following that was the evolution of the head, where we've got sensing the environment, processing information. And located in there is the cerebral ganglion, or the brain, which is a large mass of neurons responsible for sending and receiving information to and from the body. So a major integrating center here in the brain, which is concentrated in the head, which again is a very important adaptation. Very good to have that level of complexity. Okay, so we talked about the muscular system, kind of how it started. We talked about the nervous system. Now let's look at kind of what's going on inside in the gut, right, and the body plan. So when we look at a basic bilateral animal, there's, and use the worm kind of as an example in your mind here, because it's the easiest one to picture, so there's not a whole lot going on in there, but it's basically a tube within a tube. We call that the coelom. So in the coelom of these animals, some animals have a coelom, some don't. The inner tube is the gut with the mouth on one end, the anus at the other. The outer tube has the nervous system around it and the skin. And in between the inner tube and the outer tube, you've got the mesoderm, which has the muscles and in between forms the muscles and the organs, right? So the coelom is basically just a space, a body cavity between those tubes. And what it does is it provides oxygen to move and nutrients to circulate. It enables the internal organs to move independently of each other, right? And you can see here in the annelid worm, we've got our inner tube here, right? The muscles and organs derived from the mesoderm. We've got the outer tube here, skin and nervous system, and then we've got this space. And that space is the coelom. And some animals don't even have this space at all, which we're going to see, and some have sort of a space, but not really, and it's lined with different things. So we call bilateral organisms whose coelom is completely lined with mesoderm on the inside and the outside. So the gut is lined with mesoderm, and the outer skin is also lined with mesoderm on the inside. That's a true coelomate. All right. So we see that in for the most part, the most complex of the animals are coelomates. Some animals have no coelom, such as flatworms, platyhelminths, and we call them acoelomates, so there's no space in there. It's just, the gut, the mesoderm, and the ectoderm, skin, muscles, gut, no space. So some have sort of a fake coelom, bilateral organisms whose coelom is only partially lined with mesoderm. So it may have mesoderm on the right hand side of the skin, and there's a space, but there's no mesoderm that lines the gut. Essentially, that's a pseudo-coelomate. Right? It's a false cavity form. It's not a true coelom because it doesn't have the lining of mesoderm inside of both. So I think it makes more sense when you look at a picture. So let's look at a true coelom. We've got the gut here. Focus on the gut in the center in yellow. And if you look at the gut, and then you've got the skin out here. Okay. And then inside, we've got this space, that's the coelom. And notice that the gut, as well as the skin, is lined with mesoderm. Jump down here, you can see that we've got, again, a space, but there's no mesoderm lining the gut. It's only lining the skin. So that's not a true coelom. It's only partially lined with mesoderm, so it doesn't have the same functional capabilities. You can't contract as easily the gut here. Whereas an acoelomate has no opening at all. Okay. So we'll spend a lot of time looking at this, I believe, in the next couple of chapters when we focus on the protostomes and the deuterostomes. But basically, the names, so we can further break down animals into two groups. So we've looked now at various other groups, but we can look at two big groups, protostomes and deuterostomes. So when we look at protostomes, these are going to be organisms, so literally means first mouth, named for the embryonic development of the mouth before the anus. So in organisms that are protostomes, in their embryonic germ layer gastrulation, when the cells move, they're going to create, the pore first, that's going to open up and become the mouth before the anus. And then deuterostomes, which literally means second mouth, named for the embryonic development of the anus before the mouth. So it just has to do with the development here. So pro means before, so just remember mouth before anus. Deuterostomes means second mouth, or deutero means, second, named for the embryonic development of the anus before the mouth. So just make sure you keep that straight. If we focus on protostomes specifically, we can further break down protostomes into two groups, the Lophotrochozoans and the Ecdysozoans. The Lophotrochozoans, mollusks, annelid worms, flatworms, These guys are going to keep growing continuously when conditions are good. Okay, and we're going to look at all the characteristics of these again in the next chapter. This is just the basics. Ecdysozoans are your arthropods, your nematodes. So these are things that are going to molt, organs that are going to molt, like crayfish. They're going to shed their external skeletons and outer coverings when they grow. All right, so it has to do with growth there, how we're describing it. Again, there's other things we're going to see with that when it comes to protostomes. And we're going to see that the vast majority of animal species are going to be protostomes. Segmentation is something that we see in worms as being a defining characteristic. So it's the division of the body or a part of the body into a series of similar structures. We also see this in vertebrates with respect to the segmented backbone. And the segmented backbone is a defining characteristic here of vertebrates. And vertebrates fall under the monophyletic lineage, within the phylum Chordata. So in the phylum Chordata, that's where you find all the animals that you generally think of as being animals. The ones that you usually don't think about as being animals are your invertebrates. They're a paraphyletic group, and their segmentation is conspicuous in the annelids, the segmented worms, and also in arthropods, right? In insects, you see that segmentation very obvious. So the mouth, the brain, very important. Concentration of sensory organs. So I said that that's something that's really going to be important and very advantageous, I should say. A whole lot comes with having a brain, so you have a whole lot more sensory abilities when you have a concentration of neurons in one spot. So the senses of sight, hearing, smell, taste, and touch are common among most animals. And animals also have the ability to feel temperature changes. So here's just some basic examples of how animals use their various senses. Right, so fairly straightforward here. Sight, the stimulus obviously for sight is light, so look at the size of those eyes on the flies there. Hearing, obviously the stimulus is sound. Bats actually use their hearing and their sense of echolocation to determine where they are. So they're not seeing, but they're hearing to know where they are. Taste and smell, so different molecules are going to initiate that. So the binding of molecules to receptors initiates olfaction or smell or gustation. And taste, male moths have elaborate antennae to detect chemical signals in the air. And then also touch, right? Stimulus, contact, pressure. So the sea anemones, they don't move, but they do feel and detect prey as it comes by. And there's other senses there as well, kind of interesting to take a look at. Thermal energy, temperature. Sea turtles can actually sense magnetic fields. Sharks can sense electrical fields. Birds can adjust their flight patterns based on barometric pressure. And comb jellies are susceptible to gravity and can sense that, so and can adjust their body movements accordingly. All right. So this is where I'm going to end part 1 of chapter 30.